Science. 8. Genetics and Genomics. Genetics and Genomics, Decoding the Secrets of Heredity. 1. DNA structure, unraveling the blueprint of life. Understanding the double helix structure of DNA provides insights into the molecular basis of heredity. DNA, or deoxyribonucleic acid, is a complex molecule composed of nucleotide units arranged in a double-stranded helical structure. Each nucleotide consists of a sugar molecule, deoxyribose, a phosphate group, and one of four nitrogenous bases: adenine, A, thymine, T, cytosine, C, and guanine, G. The complementary pairing of these bases, A with T and C with G, forms the rungs of the DNA ladder, providing the foundation for DNA replication and the transmission of genetic information from one generation to the next. Understanding the structure of DNA has revolutionized biology, serving as the blueprint of life and providing a framework for studying genetics and genomics. 2. Genes. The blueprint for life's instructions. Genes are the fundamental units of heredity, carrying instructions for building and maintaining an organism. Each gene is a specific sequence of DNA that contains the information necessary for the synthesis of a particular protein. Proteins are involved in nearly every aspect of cellular function and are crucial for the structure, function, and regulation of cells and tissues. Genes determine an organism's traits, such as eye color, blood type, and susceptibility to diseases. They provide the instructions for the production of proteins through a process called gene expression, where DNA is transcribed into messenger RNA, mRNA, and then translated into proteins. The study of genes and their functions helps unravel the complexity of life and provides insights into the mechanisms underlying inheritance and genetic disorders. 3. Genetic code. Decoding the language of life. The genetic code is a set of rules used by cells to translate DNA sequences into proteins, the workhorses of life. It is a universal code shared by all living organisms, from bacteria to humans. The code is based on the sequence of nucleotides in DNA or mRNA, which is read in groups of 3 nucleotides called codons. Each codon corresponds to a specific amino acid or a stop signal, indicating the termination of protein synthesis. The genetic code allows the precise translation of genetic information into the sequence of amino acids that make up proteins, enabling the diverse functions and structures essential for life. Decoding the genetic code has been a fundamental achievement in genetics, facilitating the understanding of how genetic information is processed and translated into functional molecules. 4. Genome sequencing. Unlocking the blueprint of life. Genome sequencing is the process of determining the complete DNA sequence of an organism, revealing the full set of genes and providing a blueprint of life. Technological advancements have made genome sequencing faster, more accurate, and more affordable, revolutionizing the field of genomics. The Human Genome Project, a groundbreaking international endeavor, successfully mapped the entire human genome, marking a major milestone in understanding human biology. Genome sequencing has expanded to include other organisms, ranging from model organisms like mice and fruit flies to bacteria, plants, and viruses. By deciphering the genetic code of various organisms, scientists can explore the relationship between genes and traits, identify genetic variations associated with diseases, and uncover the microevolutionary history of variants inside a species, or genus or taxonomic family. Genome sequencing has transformed our understanding of genetics and opened up new possibilities for personalized medicine and precision healthcare. 5. Human Genome Project. A milestone in genetic exploration. The Human Genome Project, HGP, was an international scientific research effort that aimed to map and sequence the entire human genome, providing a comprehensive understanding of human genetics. This monumental project, completed in 2003, involved collaboration among researchers worldwide and marked a significant milestone in genetics. The HGP revealed that the human genome contains approximately 3 billion base pairs and about 20,000–25,000 genes. It provided insights into human genetic diversity, the organization of the genome, and the functions of different genes. The knowledge gained from the HGP has revolutionized various fields, including medicine, biology, and anthropology. It has paved the way for advances in personalized medicine, genetic diagnostics, and our understanding of human evolution and ancestry. The Human Genome Project stands as a landmark achievement in deciphering the blueprint of human life and continues to shape our understanding of genetics and genomics. 6. Genetic variation. Genetic variation refers to the differences in DNA sequences among individuals of a species. These variations play a significant role in shaping the diversity seen in populations, as they can influence traits, behaviors, and disease susceptibility. Genetic variation arises from various sources, such as mutations, genetic recombination during sexual reproduction, and gene flow between populations. By studying and understanding genetic variation, scientists gain insights into microevolutionary processes, population dynamics, and the genetic basis of complex traits. 7. Genetic disorders. Genetic disorders are conditions caused by changes or mutations in one or more genes. These mutations can lead to abnormalities in the structure or function of proteins, affecting various aspects of an individual's health. Examples of genetic disorders include cystic fibrosis, sickle cell disease, Huntington's disease, and Down syndrome. Studying genetic disorders not only helps in understanding the underlying mechanisms of these diseases but also provides opportunities for developing targeted therapies and genetic counseling to individuals and families affected by these conditions. 8. Genetic inheritance. Genetic inheritance refers to the process by which traits, including physical characteristics and susceptibility to certain diseases, are passed from parents to offspring. The principles of inheritance were first described by Gregor Mendel, who established the fundamental laws of genetics. Understanding genetic inheritance allows scientists to predict the likelihood of certain traits or disorders in offspring based on the genetic makeup of their parents. It also plays a crucial role in comprehending the genetic diversity within populations and the mechanisms that contribute to evolutionary change. 9. Epigenetics. Epigenetics is the study of heritable changes in gene expression that are not caused by alterations in the underlying DNA sequence. These changes can be influenced by environmental factors, lifestyle choices, and other external factors. Epigenetic modifications, such as DNA methylation and histone modifications, can impact gene expression, turning genes on or off without altering the DNA sequence itself. Epigenetic mechanisms play a crucial role in development, aging, and the response of organisms to their environment. Understanding epigenetics provides insights into how genes are regulated and how environmental factors can influence gene expression and phenotype. 10. Pharmacogenomics. Pharmacogenomics is an interdisciplinary field that combines genetics and pharmacology to personalize medical treatments based on an individual's genetic makeup. By analyzing an individual's genetic variants, particularly in genes involved in drug metabolism and response, healthcare professionals can predict how a person will respond to specific medications. This knowledge allows for more targeted and effective treatments, minimizing adverse drug reactions and optimizing therapeutic outcomes. Pharmacogenomics has the potential to revolutionize medicine by tailoring drug prescriptions to individual patients, improving patient safety and treatment efficacy. 11. Genome editing. Genome editing refers to the precise modification of an organism's DNA using techniques like CRISPR-Cas9. This revolutionary technology enables scientists to edit, add, or delete specific sequences within the genome, offering tremendous potential for treating genetic diseases and advancing our understanding of gene function. Genome editing has the power to correct genetic mutations, potentially curing inherited disorders. It also allows researchers to study the effects of specific genetic modifications, accelerating the development of novel therapeutics and the investigation of gene functions. 12. Comparative genomics. Comparative genomics is the field of study that involves comparing and analyzing the genomes of different species to understand microevolutionary relationships, identify conserved regions, and explore variations in gene content and organization. By comparing genomes, scientists can uncover shared local, not universal, ancestry, identify genes and regions responsible for specific traits, and gain insights into the microevolutionary processes that have shaped life on Earth. Comparative genomics has revealed fascinating connections between species and provided valuable information about gene function and genome microevolution. 13. Functional genomics. Functional genomics focuses on understanding the function and regulation of genes and their products, such as proteins and non-coding RNAs, on a genome-wide scale. This field encompasses various experimental and computational approaches to identify and characterize the roles of genes and their interactions within cellular processes and biological systems. By studying gene expression patterns, protein-protein interactions, and the impact of genetic variants on gene function, functional genomics provides critical insights into the complexities of biological systems and the underlying mechanisms of diseases. 14. Population genetics. Population genetics examines the genetic variations within and between populations. It investigates how genetic traits are distributed, how they change over time, and the forces that drive these changes, such as natural selection, genetic drift, and gene flow. Population genetics provides valuable insights into microevolutionary processes, the origins of biodiversity, and the genetic factors influencing population dynamics. By studying population genetics, scientists can better understand the genetic basis of adaptation, speciation, and the maintenance of genetic diversity. 15. Molecular genetics. Molecular genetics is the branch of genetics that focuses on the study of genes and the molecular processes involved in their structure, function, and regulation. It encompasses the investigation of DNA replication, transcription, translation, gene expression, and the interactions between genes and their products. Molecular genetics provides detailed insights into the mechanisms of gene action and the molecular basis of genetic disorders. This field has been instrumental in unraveling the intricate workings of DNA, RNA, and proteins, paving the way for advancements in genetic engineering, diagnostics, and therapeutics. 16. Cancer genomics. Cancer genomics explores the genetic changes and alterations that occur in cancer cells. By studying the genomic landscapes of various types of cancer, scientists aim to identify the genetic drivers of tumorigenesis, understand the mechanisms of cancer progression, and develop targeted therapies. Cancer genomics has revealed the diverse genetic profiles of different cancers and provided critical knowledge for precision medicine approaches. It plays a crucial role in identifying genetic markers for early detection, predicting treatment responses, and uncovering potential vulnerabilities in cancer cells. 17. Prenatal genetic testing. Prenatal genetic testing involves screening or diagnostic tests performed during pregnancy to assess the genetic health of the fetus. These tests can detect chromosomal abnormalities, such as Down syndrome or Edwards syndrome, as well as specific genetic disorders. Prenatal genetic testing provides valuable information to expecting parents, allowing them to make informed decisions regarding pregnancy management and potential treatment options. It offers insights into the genetic health of the fetus and helps identify conditions that may require medical interventions or supportive care after birth. 18. Behavioral genetics. Behavioral genetics investigates the genetic factors that contribute to behavioral traits, including both normal variations and behavioral disorders. By studying the genetic underpinnings of behaviors, such as intelligence, personality traits, mental health disorders, and addiction, scientists aim to understand how genes and the environment interact to shape individual differences. Behavioral genetics provides insights into the complex interplay between genetic and environmental influences on behavior, shedding light on the nature versus nurture debate and contributing to our understanding of human psychology and behavior. 19. Bioinformatics in genomics. Bioinformatics plays a critical role in genomics by providing computational tools and methods to manage, analyze, interpret, and visualize large-scale genomic data. The field of bioinformatics combines biology, computer science, and statistics to handle the vast amounts of genomic information generated by high-throughput sequencing technologies. Bioinformatics tools enable the identification of genes, the comparison of genomes, the prediction of protein functions, and the exploration of genetic variations. By leveraging bioinformatics, researchers can extract meaningful insights from genomic data and uncover new knowledge about genes, pathways, and evolutionary relationships. 20. Genomic medicine. Genomic medicine integrates genomic information into medical practice, aiming to enhance disease diagnosis, treatment, and prevention. By analyzing an individual's genome, healthcare professionals can gain insights into their susceptibility to certain diseases, predict medication responses, and tailor treatment plans accordingly. Genomic medicine has the potential to revolutionize healthcare by enabling personalized and precision medicine approaches. It offers the promise of improved diagnostics, targeted therapies, and preventive measures based on an individual's unique genetic profile, leading to more effective and personalized healthcare outcomes. 21. Gene therapy. Gene therapy is an experimental technique that involves the delivery of functional genes or genetic material to treat or prevent diseases. By introducing therapeutic genes into a patient's cells, gene therapy aims to correct genetic mutations, replace faulty genes, or modulate gene expression. This approach holds promise for treating a wide range of genetic disorders, including inherited conditions and acquired diseases. Gene therapy has the potential to provide long-term or permanent solutions by addressing the underlying genetic causes of diseases rather than just managing their symptoms. This represents an exciting frontier in medicine with the potential to transform the treatment of currently incurable conditions. Thanks for watching. Please like, share and subscribe, and comment below if you agree, or would like to add your thoughts.